Good afternoon, everyone. Let's get started. Thank you all for coming to our next seminar, series speaker. Today we have the distinct privilege of introducing my colleague, Mitesh Borad from the Mayo Clinic. He's an associate professor of medicine and an oncologist at the Mayo Clinic, Arizona. He's the director of the liver and biliary cancer research program at the Mayo, as well as director of the, as we can see, director of the GI Cancer Gene Therapy and Oncolytic Virotherapy Lab, also at the Mayo Clinic. He's a member of the Center for Individualized Medicine, which we'll see introduced into his talk today. He has a prestigious academic background. He got his BS in biomedical engineering at Boston University, MD at the University of New Jersey, resident at Cedars-Sinai Hospital, medical oncology fellow at Tulane, and I assume acquired part of his molecular training as a research scholar at TGen University. He's a recipient of a new Innovator Award from NIH, a very distinguished and actually highly competitive award, as well as a Paul Calabrese Scholar in Clinical Translational Research, again, which I think we'll see as characteristic in his work and what he'll describe today. He's currently vice chair of the International Cholangiocarcinoma Group, as well as a standing member, again, those of you who are looking for submitting applications, a standing member of the NIH Drug Discovery Study Section. Always good to have a friend in the family along with the study section. So, as we'll see, his broad interest in phase 1 drug development, including first-in-human trials for oncolytic virus therapy, with interest, as his titles would suggest, in pancreatic cancer, biliary cancer, liver cancer, and other rare cancers, as well as identifying and generating genomic and novel new clinical trials therapies. So today, he's going to be speaking, as his title indicates, from targets to translation, deconvoluting tumor vulnerabilities in drug development in hepatobiliary cancer. So, over to you.
Okay, thank you, Ken. I appreciate the very, very kind introduction, and I'm excited to be here with everyone today. Thank Dr. Nessie and Melissa, who I've met on a few occasions before, to be amongst a very eclectic group, as Ken has put, with a very interesting concept in terms of how the center has been organized. So as Ken mentioned, I'm a medical oncologist. I have decided to focus on cancers of the gastrointestinal system, particularly those of the liver and biliary tract, because these are amongst the most lethal and where there's considerable opportunity to try to make a change to help patients. So I will be using these two cancers as examples of how one can conceptualize how to tackle a problem in terms of going from not knowing much to learning quite a bit, hopefully, and making improvements on a therapeutic standpoint. Okay, so again, thank you so much for this wonderful opportunity to come here and speak to you. Given that I work quite a bit with industry and conducting trials with recent New York Times visibility, I want to make sure that these are some of the companies I'm working with in terms of conducting trials for some of the targets I'll describe. So for those of you who may have had the opportunity to interface with Mayo Clinic here in the region, Mayo Clinic has been here around 30 years, and ASU and Mayo Clinic have had a tremendous collaboration over the years at all levels, including research, with the medical school education, and of course with many of the efforts involving ultimately patient care. So people are very familiar with the concept of bench to bedside research, and what I'll highlight here is sort of the opposite of that concept, which would be starting at the bedside. So starting with the patient or humans as the model system and then working your way backwards, as opposed to starting in, say, cell lines and then trying to take that to patients. So the first concept I'll go over is what we would call sort of precision medicine broadly, or targeted therapies. This concept is now popular, but it's not entirely new. In fact, this magic bullet concept was being explored since the days of Ehrlich, where they were actually looking for drugs for syphilis back in the day. And the types of magic bullets we're looking for have changed, but the concept remains. So the breakthrough of this arena of drug discovery and development could be, say, traced back to imatinib or Gleevec. And this is a drug that is used in a type of leukemia called chronic myelogenous leukemia, which was incurable before these drugs came out. And after these drugs started being used, the vast majority of patients essentially are able to stay on these drugs for 1 to 2 decades, and many of them would be considered cured. So you went from incurable, understanding what drove this, to now these patients having close to normal lives. So that led to a lot of excitement, and people felt we were only a few steps away from solving most of the things that look insurmountable in the cancer arena, and that we just needed sort of the rest of the magic bullets and we'll be there. But unfortunately, what happened with Gleevec was a perfect storm in terms of the target being the right one, finding it at the earliest stages in the cancer, and the drugs being not too toxic and working spectacularly. And this was not really replicated to that degree until some recent times. So I'll use cholangiocarcinoma, which is a cancer of the bile ducts, as an example of how precision medicine can potentially make a difference when you just start with the concept and start applying it. So this is a cancer that, as I described, occurs in the bile ducts, which are tubes in the liver. They help transport bile, which the liver makes. And then the gallbladder, which is a balloon-shaped organ in the liver, helps store the bile. And the cancer can arise in any of the places where these tubes are, whether they're inside the liver or as they make their way outside. When you look under the microscope, you see that besides the cancer cells, which are shown in brown there, there's a mixture of both living and non-living elements. So there's really sort of an ecosystem that exists, and we shouldn't really think of cancer as just those brown entities, but how all the different entities communicate with each other. So you have immune cells, stellate cells, fibroblasts, and then you have acellular material comprised of things like collagen and hyaluronic acid and other things, and how all of this works together, and understanding how that could change when you change things in one or the other can really improve our understanding. And I think groups such as what have been put together here can really tackle this problem in the best way, as opposed to sort of single expertise groups that we usually work with. So in the last decade or so, with the advent of next generation sequencing, the ability to genetically characterize, or as we say, profile cancers has improved considerably. And here's one of the early efforts that I was involved with when I was at TGen, where we did profiling, but it was really a more poor man's profiling with things like immunohistochemistries and DNA microarrays, where we looked at expression of genes, not at the level we're doing now, really. So now we have more powerful technologies where we could look at whole genomes or whole exomes, and this has been applied to the vast majority of cancers at this stage with national and international efforts such as the Cancer Genome Atlas and International Cancer Genome Consortia. So in this rare cancer even, here's an early effort where they did exome sequencing using just one platform and found a few abnormalities to start understanding what this cancer entails. However, I think the important thing to realize is that just one platform doesn't tell you the whole story sometimes, and you also have transcriptomes, you have epigenomes, you have other elements, and when you put the data together, you really get a better sense of what the cancer is. And I like to use the elephant and blind man example to help illustrate why just one platform doesn't give you the full picture. You might make very limited conclusions about things when you just use a single platform, but if you start putting them all together, you'll really know the entity you're studying. So in the study we had done as a collaboration with TGen and Mayo Clinic, looking at applying integrated sequencing to patient care in a real time. We had this sort of workflow where we would get tissue, we would extract analyte, sequence it, get the results back, interpret it, and see if we could apply it to patient care all within just several weeks. So given how long it took to sequence the first genomes, to be able to do this in a routine fashion represented one of the first efforts. So this was about 9 years ago, and I think the pancreatic cancer genome that we sequenced was the second cancer genome ever sequenced at a whole genome level. Okay. So when you do integrated sequencing, you have the ability to inform yourself of many, many different aspects. So people focus usually on therapy selection, but because you're looking at all aspects, you can look at what's in the germline and thereby convey risk to family members if it was a germline event. You can see how drugs are metabolized. You can look at prognosis if it's not in the context of needing to do therapy. The last two pieces could be things that are of particular interest to centers like this one, where this is not a static process. As things evolve with therapy interventions, you might have genomes that change in the tumor, and you want to go back and reanalyze them to see if there's changes in the clonality, mechanisms of resistance, and you can use more sophisticated assays like liquid biopsies to also monitor the response. So again, evaluating the genome on a real-time basis. So as I showed you before, some things fall within the clinical realm, some in the research realm. We're hoping that as we work out the kinks, that it's all really just in the clinical realm. Research becomes practice, as one would say. So the challenges at the time were, well, how fast can you get the results? You get a lot of data back from these types of analysis. How do you know what are the important things, what we call the drivers, and what are sort of just passenger events? What do you do with incidental findings? Where you biopsy and what results you get back, how does that matter? So that's tumor heterogeneity. Many of these challenges still remain, but it's important to be cognizant of these. Okay, so one approach is where you do parallel processing, where you do things that you can get results back quickly, and if there's something you can act upon, you act at a sort of F16 level, and then if things can come back a little bit more reasonably, you act at the Ferrari level, and then you get the tortoise level. But there could be something there that's very important, so if the patient is able to make it that far, you can still use that information, right? So parallel processing. Okay, so finding what's important. So there's the known knowns, the unknown unknowns, etc., right? When you get the data back, how do you determine what's important? So you can apply some principles, and I'll show you some of these. So here's the ones that I use. So if you use multiple platforms, if you get concordance, I feel that helps elevate something to a driver. If you see something where in the transcriptome data, you get dominance of an event, what we term mutant specific expression, that might be an important event. The third one is somebody felt that was the case. So what folks at Harvard usually term heuristic knowledge. I don't think there's a way to characterize that other than expertise and just a sort of gut feeling, right? With mutations, you can't rank events. However, with copy number changes, you can rank them. So it could be 2 copies or 10 copies or 20 copies. So one would think that 20 copies would maybe be more important than 2 copies. So ranking can help with those. And then if you have a pathway and you find that there's multiple things happening in that same pathway, that could be a way of saying these all collectively form a driving event. So we'll talk about, I often get confused with this guy just because of everything but one letter, right? He isn't a cousin of mine, by the way, but okay. So here's a case with a cholangiocarcinoma where we did multi-platform sequencing. We found a fusion event between the genes Fgfr2, which is fibroblast growth factor receptor 2, and MGEA5. And this was present both in the transcriptome data and the exome data. So we knew it was both a DNA and RNA level event. It was being expressed. So this would be termed multi-platform concordance. And because this is happening in a kinase, there's a high chance that something functional would be occurring. So we didn't have any further functional data when we found this in the patient. We had to make a decision, and we went straight to application. So we treated this patient with an inhibitor of fibroblast growth factor receptor pathway, which is ponatinib. It was a sort of repurposed use. And we saw that this patient had shrinkage of the lesions in various parts of their body. So this conveyed to us that this may be an important driver event, and we were able to do this by skipping many of the steps, right? So you'd usually start with cell lines, do functional studies, animal studies, toxicology studies, and then go to the patients. Here, we went straight from possible data to knowledge and then to patient application. And of course, it was a guess, right? But we're able to test it out quickly in the human model system. We tried this in several different patients, and we saw the same results. So we had a good sense that this was a real event. A number of other groups confirmed these findings. And at that stage, we said, this is really a key driver event. So rather than spending a lot of time deciding whether this should be something important or not, we went on sort of a more comprehensive approach where we said, okay, now we know the target. It's been discovered. Let's see if we can develop a diagnostic. Let's see if we can find an animal model. Let's see if we can generate cell line models. Let's do larger clinical trials, and let's understand the target biology. And then using this framework has led to additional understanding of the pathway, even beyond what was already known. So here's the pathway. The gene and protein is a receptor tyrosine kinase, where you get expression, including at the surface of the cell. The protein components dimerize, and the dimerization, which is fueled by binding of the ligand to the receptor, creating the dimerization, then fuels the subsequent elements of the cascade, where you get a scaffold, which is this FRS2 that you see in blue there. And that brings upon further events which lead to cell proliferation, invasion, metastasis, what we term the hallmarks of cancer. So the event starting at the top leads to all the events at the bottom. So if you affect things at the top or at the bottom, you can get effect. But ideally, you want to start at the top so you get specificity. Okay. So then we said, well, what is the prevalence of this event? We had only done a few cases, and we broadened our efforts, and others did the same. And we found that this was found at about a 15% frequency, mostly in patients with bile duct cancer in the liver. So intrahepatic cholangiocarcinoma, 15% frequency. How could you quickly diagnose it? So there's a a well-known technique that many of you may have used called fluorescent in situ hybridization, based on the knowledge of where the break occurs to create the fusion. We found it was a very small region of several kilobases, and we work with our colleagues in laboratory medicine to develop a FISH assay where you can create what's called a break apart assay, where if you have the fusion, you see the separation of the signals with red and green here, and if you don't, you see a yellow, but you don't see any yellow here. So this case has has the fusion and is a positive example. And within 2 years of when we first described it, we had a diagnostic, right? Very, very quickly. Here's the pathway again. And here, I think the important thing to understand would be, while you might have a protein you're talking about, really you're talking about many different proteins when you expand your definition to isoforms. And where the break was occurring, this was leading to the creation of only one isoform. So there's an FGFR2 3b isoform and a 3c isoform. When you get the fusion, because of how alternate splicing would work and what you would need to create one or the other, you could only go to 3b. So now you're getting further into understanding how you can go after this target by going after an isoform. So different ligands bind to different isoforms, and now you have a better understanding also of what could be the etiology of these cancers, because obviously it's not going to be the ones on the the right there, it's going to be the ones on the left that are going to contribute, because you only have a single isoform that's found in all these cancers. Animal models. So a couple of my colleagues who are featured here, Lewis Roberts and Mark Trudy, who are in the Rochester campus at Mayo Clinic, had been over the last few years generating patient-derived xenograft models where they would take human tissue and implant it into immunocompromised mice and grown a series of these cancers. So once we found the fusions, I chatted with them and said, why don't we do RNA-seq on a bunch of your cases? And they said, go ahead, and we found one that had the fusion. So now we had a model system that we found after we found the first case in several patients and treated them. So what's the advantage of having a model system? Well, it's a representation of the human cancer that lives on that you can perturb. You can do karyotyping. You can do the diagnostic again, like shown here. We treated this xenograft with ponatinib, which we had used in the patients, and we found that it worked the same way. But we also saw that it wasn't that much more effective than a standard therapy. So we got some sense that we may not be able to beat standard therapies, but we may be able to do something with less side effects. We also studied several different inhibitors of the FGFR pathway, and we found that more specific inhibitors, one is called BGJ398, I don't know if it's that visible in the upper left-hand corner there, that was the most effective. And then we were able to do sort of more functional studies validating what we had seen in patients, but here we can get more detail in terms of what we would want to evaluate. Okay, so moving on, I'll show another case where further knowledge was generated with additional study of this pathway. So again, we started with a patient. This is a patient who had received standard therapy. We did next generation sequencing on the patient. The patient was started on an FGFR inhibitor. The patient had a number of mutations which are found in many cancers, such as P53 mutation. Here's another mutation of unknown significance that we had found in another kinase. And we found a mutation, not a fusion, in FGFR2 at position 276 from F to C. So it turns out that that position is where some of the dimerization of the receptors occurs, and it would potentially functionally affect dimerization and make it more potent. So it turns out that this, when treated with the inhibitor, actually had a response. So now we were able to put together the structure, functional, and clinical data. And now if another patient shows up with that mutation, we know that that is someone we would want to also treat with these inhibitors. So this continuum of taking what we find from patients with model systems, integrating it and converting data into knowledge is the platform that we feel is of most value. When we did further functional studies, we saw exactly what was seen in the patient. This is a more proliferative mutation. And collectively, these efforts have led to a number of clinical trials with both small molecules and antibodies. The one on the top is the antibody using the concept that I described to you, and some of the other ones are other small molecules that can go after the protein. I mentioned to you that BGJ398 was the most potent of the selective inhibitors, so we conducted a phase 2 study that was larger in these patients who had the fusions. This is a way of representing what happened to the patients, what we call a waterfall plot. And you can see that most of the patients had some level of tumor shrinkage and are all represented on the right, and then a few patients didn't respond at all. Diagnostic methods can also utilize some of this type of knowledge. So you can do a standard CT scan, but can you extract more from these images other than what the eye can see? So we work with actually colleagues here at ASU, and you may recognize some of the names: Teresa Wu, Yinlin Fu, and Mao He, who develop machine learning algorithms to be applied to CT scans to patients who have these fusions and who don't. And we were able to, or rather, they were able to come up with an approach where you could maybe identify a patient who had the fusion versus those who didn't. So quick diagnostic using just an image. And using Fourier transform-based methods, the case on the right that's circled, you can see that it's quite distinct from the other cases. And since these early efforts, we've expanded to see how accurate these methods are, and they're about 80 to 90% sensitive in detecting cases. So again, from that first discovery, you can apply all the other avenues that are available, from diagnostics to therapeutics to further model systems, to really expand the work. So target discovery is very, very key. A few of my colleagues here who have developed liquid biopsy techniques are working on techniques to see, can we detect these in the blood, and can we follow the response to treatment if we do these things. Something that could be of interest to the group here. Zebrafish, when exposed to chemicals, actually get a lot of bile duct cancer, so could we use zebrafish as model systems for these kinds of things? Okay. If we look at another method in terms of who's the driver, if you have excess expression in the transcriptome, that could be a key. So here's another patient that we did in that series I showed you, where we found a mutation in a gene called ERRFI1. So ERRFI1 is a negative regulator of the epidermal growth factor receptor pathway, which is a proliferative pathway. So think about losing a brake. So an inactivating mutation in this gene would leave proliferative pathways unchecked. A number of studies had been done before where they studied this protein and suggested it's tumor suppressive and negative growth effects. And they found that mice that didn't have ERRFI1 actually developed bile duct cancers and bile duct adenomas. So now seeing this in the patient, put it all together, and just again from patient-based discovery, we were able to say this is a relevant gene in human cancers too. So like we did before, we just went ahead and treated the patient. And here's a PET scan showing the tumor lighting up significantly. And after a few months, you can see most of the tumor is shrunk and gone. So now we know that this gene is also a driver based on these types of things. Here's another case, and this is the last one I'll show before I move on, where we did sequencing as well. And here's the usual heuristic approach we would follow. So I talked to Melissa a little bit about this, where you would say you have data, and the key is to convert it to knowledge, and then once it's knowledge, then if you see it again, you know what to do, right? So it's just the first or second time that you need to go through it that you need to figure out what it means. So you see something, in our case, whether it's data or it's going to become knowledge, the intervention is the drug, and we see what happens. Someone else put it much more elegantly than I can put it, where, is this in the unknown unknowns category or the known knowns, or, unknown knowns, or, who knows? So in this case, what we saw was another fusion of a gene called NRG1, which is neuregulin, and we saw it in 2 platforms. And it turns out that NRG1 is a ligand to 2 receptors, particularly the HER3 receptor, which dimerizes with other HER receptors, and that could drive the pathway. So based on the known biology of NRG1, we surmised that this is an important event. We treated the patient with drugs that interfere with HER2 signaling, which is one of the downstream events. And you can see that they had a massive tumor to start with, and just 8 weeks later, almost all of it's gone, right? So I'm showing you obviously all the cases where it worked and leaving out the so don't get the impression that this works all the time. Okay, so anyway, the patient had a dramatic response. Interestingly, what we saw in this patient with regards to evolution of the tumor was the patient progressed, and we resequenced, and we saw an amplification in another gene called EGFR. So there were probably 2 clones, and we eliminated one, and we saw the other, right? So this is seen in very rapid fashion. This happened after only 4 weeks. So clonal evolution of tumors can be extremely rapid, and this is the importance of analyzing several times as you go through it. So we saw that the original event was still there, but there was a clone that also had the amplification, so an added advantage. And this was all seen in the whole genome data, so I'm just showing you how we found it. So the conclusions from what I showed you here was that starting with the patient, using a technology that's pretty robust, you can discover things. Once you discover them, you can take them back to your platform and make additional discovery, and you can take it back to the patients, and you can keep going with this process. So you need multidisciplinary teams, and these types of groups that you guys have assembled can add to these sorts of efforts. And this is what that paradigm would look like. You start with the patient at the middle, and you just do everything I described. Okay, so obviously there's a lot of work to do. This is actually in Arizona, if you ever, when you're coming back to Phoenix. Okay, so I'll move on to hepatocellular cancer, and here I'll switch gears considerably in terms of what therapeutic platform we use. So this is now cancer of the liver itself, not the tubes inside it. It's the most common primary cancer of the liver. The causes are hepatitis viruses, fatty liver disease, increasingly now alcohol abuse, and then rare syndromes. In the US, it's quite uncommon, about 40 to 50,000 cases, although it's growing at a very rapid clip, unfortunately. Worldwide, this is a much bigger problem with almost 1 million cases, mostly in low-income countries. Sorafenib is a drug that targets blood vessel growth through kinase inhibition. And these are the survival curves for these patients. So you can see in yellow that it helps, but all the patients die. So there's considerable need to find sort of game-changing ways to treat these patients. So there's an unmet need, as I described. One of the ways we figured we would tackle this is through using recombinant viruses, which I'll get into in a little bit. So viruses have been known since the late 1800s, and the first virus that was identified was actually a plant virus, tobacco mosaic virus. I think there's people here at ASU that work on tobacco plants, at least, for various things. And interestingly, since around the time that the viruses were discovered, a number of observations had been made over about an 80-year period where people who had been exposed to viruses suddenly had their cancers go away. And in the 1940s through 70s, attempts were made where people intentionally introduced viruses to try to replicate what was seen in these instances. And they found that they worked in some cases. Unfortunately, the idea was a little bit ahead of its time, and wild-type viruses, while they may help with cancer, they also cause illnesses. So, you introduce hepatitis B in somebody. I mean, you're going to bring about problems with yourself, and you give somebody mumps. I mean, if they're vaccinated, good, but if they're not, you're going to give them disease, right? So as more understanding of these organisms grew, or these particles, or between the interface of live and unlive, we began to gain an understanding of how we could apply them in a more sophisticated way, and we started classifying them on their nucleic acid capabilities, and then also in terms of different families like adenoviridae, herpes viruses, vaccinia, which I think Dr. Jacob here works on, vesicular stomatitis virus, etc. Right? So I'll tell you a bit more about VSV, or vesicular stomatitis virus, which is one of the platforms my lab works on considerably. So as you can see, it's shaped like a bullet. Normally it causes mouth sores in animals. Cows, pigs, horses. It's transmitted by mosquitoes. I spoke to someone here about mosquitoes a little bit. And it has some endemic nature in certain parts of the world, and in others much less. In South America, some areas are endemic, and others no incidence, and in the US certain parts have had exposure and others none. But most parts have had none. So this is an advantage of this virus, where unlike, say, a cold virus, everybody might have been exposed to it and may have antibodies to it. With VSV, very few people would have exposure. So we'll go back to the magic bullet because it looks like a bullet. And here's its genome. It's an RNA virus, negative strand, very simple genome, only 5 genes that form various components of the virus. What are the advantages of this platform? It's a small genome. It can get into most cells. In fact, people use VSV pseudotyping with different viruses to try to increase the tropism to try to get different things into cells. Very low seroprevalence, so you won't get it neutralized if you use it as a therapy. It grows very quickly at the rate of 10,000 to 100,000 particles from each one in just a day. And it's quite sensitive to type 1 interferon response, and I'll show you why that's important. So again, as I showed you before, you need a whole team to really execute these concepts into proof of concept. So folks would be involved in discovery. So Glenn Barber, who's a collaborator at the University of Miami, actually found that this virus has anti-cancer capabilities. A number of my other colleagues did some of the initial preclinical work. We have a toxicology core at Mayo that can do formal toxicology studies to take these into the clinic. And we can make GMP grade vector to administer to patients, all within the framework of an academic organization. So I showed you the timeline. So VSV has been known since late 1880s. And you can see when the first patient was dosed from when knowing about VSV. And even it took 12 years from when it was known that it has anti-cancer properties to when we could apply it to the clinic. So there's a big barrier there that we hopefully can work together to remove to apply concepts to patient use as fast as possible. So here's the genome. What was done was, for the vector I'm going to show you, was the interferon gene was put between the last two genes. And the reason for putting the interferon gene in there was to take advantage of two factors. One was this virus is very sensitive to interferon, and in cancer cells, interferon pathways may be defective in a certain proportion. So even if you have the gene, it doesn't matter if you have interferon, whereas in a normal cell, which are above, it would get shut off, and in tumor cells, it would just keep replicating. So you would create a selective advantage for this vector with a safety advantage in cancers versus normal cells. When they looked at normal cells versus cancer cells, they found that the effect was primarily in the cancer cells. Normal cells did okay because they had intact type 1 interferon response. And the safety aspect is what I described, right? So a normal cell would shut it down and a cancer cell wouldn't. Now, because you're producing interferon itself would have immunomodulatory effects, so you could take advantage of it as a sort of secondary mechanism of anti-tumor effect other than just the lysis. Animal studies showed that this was effective as well. And toxicology studies were done to take this into the clinic at the right doses. So this helped determine what dose we would start at. A number of other vectors with different modifications have also been under evaluation. So the first one listed there, for example, has a mutation in the matrix protein, which also leads to higher levels of interferon. So it recapitulates some of the kinds of things I described with the vector we're using. So this is the trial I've been working on for the last decade now, since I've been at Mayo. We've just finished it. And here we would take the virus and inject it into the tumors of patients who have liver cancers. And this is the kind of needle we would use to spread the virus, where instead of just one location, when you inject it, you can spread it into multiple areas of the tumor. And in these trials, typically the objectives are see what the safety is, see what kind of serious toxicities you get. And because of the kind of drug we're using here, and it's a live replicating drug, we say, what's the sort of infectious nature of the virus? How long does it hang around in various bodily fluids like feces, saliva, blood, urine? And then, of course, because this could have many different kinds of effects, what are the clinical effects in terms of tumor shrinkage, and what are the immune effects of this kind of therapy? How quickly are antibody responses mounted against the virus? Does it activate T-cell responses in the tumor, natural killer cell responses in the tumor, et cetera, right? And then, of course, the last item is of significant interest is how does the type 1 interferon response play a role in this therapy? So these are more standard definitions on how we say something is severe or not. Anticipated toxicities were mouth sores, just like the animals I showed you. Because it's a virus, could you get a flu-like illness? This virus is a member of the rabies virus family, so would you get neurotoxicity? And I know there's faculty here at ASU who've been working on this kind of problem. Would you get a severe reaction, like a cytokine release? Because you're injecting it in the liver, would you get toxicity there? So we usually put criteria in place to make sure that we're not selecting very sick patients, but of course they're not healthy either, to make sure that there's sufficient safety when doing this. We gave the virus by injecting it in one episode. You could inject it in several parts of the tumor, but we selected one relatively large tumor to inject it in. We used an ultrasound to do this. And we studied a number of dose levels where we gave the virus. If it did okay, we went higher, et cetera, right? These are the demographic characteristics of patients that were enrolled, typical for liver cancer patients, a bit older, with the etiologies that I showed you, who had received prior therapies. What kind of side effects did we get? Many of what we expected: fevers, chills, headaches, irritation of the mouth. And we saw just with the virus alone that one patient had tumor shrinkage more than 30%. A number of other patients had tumor shrinkage, but not 30%, but lasted a considerable amount of time. And I'll show you kind of anecdotal details of some of these patients. So here's a patient with hepatitis C-related liver cancer, previously treated with standard therapies. Those didn't work. We injected the tumor where it's shown, and that area that was injected became a bit cavitary, which is shown in the dark area there. When we gave the virus, the hepatitis C level also went down. And could this have been due to interferon? Possibly, but the patient had received interferon previously, actually, and had had no effect. So perhaps related to the virus itself somehow. Here's another patient, also with hepatitis C-related liver cancer, progressed on standard therapies. On the top is the blood marker called alpha-fetoprotein, which we can measure. And if it's high, the cancer is generally worse, and if it's low, it's better. So you can see after we gave the virus for several months, it went up considerably. And then it started going down and stayed down for almost a year. So maybe there was sort of an immune-mediated effect in this patient. And this patient also had hepatitis C levels go down, both in the tumor and in the blood. Here's another patient where we injected and it shrunk considerably. Of interest, the tumor lesion that I showed that shrunk was not the one that was injected. So again, could there be an immune-mediated effect, or could the virus have spread from where we injected to the tumor that got affected? This will be the last sort of anecdote I show. Here's a patient who had an injection, very bright tumor that was injected, and then just within a couple of months, the whole tumor is dead, right? So you can have a variety of effects from direct killing potentially to delayed effects, maybe mediated by the immune system. Finally, I'll close with sort of an unfortunate case that occurred during the trial. Here's a patient that had a lot of cancer. This was a patient with colon cancer, actually, with liver lesions. And the tumor was injected. After the patient had the therapy, this is the background of the patient, had standard therapies. So several days after the therapy, started developing some shortness of breath, felt to maybe have some fluid in the lungs. The fluid was removed. We felt the patient might get better, but unfortunately he didn't. He started becoming somnolent. The liver enzymes started going up considerably. And by a week after the treatment, the patient was completely obtunded. We felt maybe the virus had something to do with this. We tried antiviral therapies. The patient started having end organ failure, including liver and kidney failure. And 2 weeks after we had started the treatment, the patient's family decided we didn't want to take any more aggressive measures, and the patient passed away. Well, what happened in this patient? So there was lymphopenia in this patient. The platelet count went down. The coagulation factors went up, all indicative of disseminated intravascular coagulation. There was possibly some evidence of tumor lysis with the phosphate levels going up and the calcium going down. All the liver enzymes considerably worsened, bilirubin, AST, ALT, and you can see that they started improving too. And I think that was when things had peaked and actually things were on the mend. When we got a biopsy of the tissue, what we saw was massive liver injury to normal liver too, not just the tumor. We look to see what, I'll go back to that patient in a little bit. We look to see what, if we could detect the virus in any of the cases, and it turns out that In all the patients before this patient, we couldn't detect the virus in any of the bodily fluids. However, in this patient, which is shown at the far right, we had significant viremia. So the virus was, even in the blood, it was producing so much virus up to a week later. So there was significant replication that was ongoing for a long period of time. Antibody responses occurred in all the patients, but a few, including the one I described, but it didn't happen until approximately 2 weeks after the virus was injected. So there was no protection for the time period when things were worsening. We took a further look at things, and what we found was we were able to recover virus in the tumor tissue, the blood, primarily, and we were able to also recover it in tumors that were not injected. So what had happened was the tumor that was injected became a viral factory. It produced lots of viral particles. They went all over the body. Normal cells didn't get affected. Other tumor cells got infected because they were susceptible, and the virus was transmitted through the blood, not via the liver itself. We looked to stain the virus in the tumor tissue and the normal tissue. We only saw it in the tumor tissue. So the virus did what it was supposed to. It infected the tumor, it replicated there. In normal tissues where type 1 interferon response was intact, it was fine. Now, if you remember, the virus had interferon beta. So the transgene became the liability. The virus replicated to hundreds of thousands to millions of copies per particle, and it produced very, very high levels of interferon beta, which eventually led to the toxicities I described. The interferon beta caused liver injury, that led to liver failure, that led to kidney failure. And I suspect if the virus didn't have interferon beta in it, the patient may have still been alive today. Okay. We also looked to see other cytokines. The main one that was elevated was the interferon beta. Nothing else was significantly changed. As I mentioned, it replicated significantly in the tumor. There was some in the non-injected tumor, but it was coming from the injected tumor by the viral factory spread. We saw this in other fluids, predominantly the blood, which was the mechanism of transport. Was the interferon coming from the virus, or was it coming from the host? So we looked at that too, and almost all the interferon was from the transgene. So we were able to do that through a SNP that was found in the interferon transgene in terms of the insert. So this was not from the patient, it was from the vector itself. We were able to use the RNA-seq data from samples taken from this patient and other patients, and see that, yes, there was quite a bit of viral transcripts found in this patient, but we also saw it at lower levels in some of the other patients, which were not detected by standard methods. So again, application of these types of technologies can be quite helpful. We looked to see what were the differences between the patient I described and other patients in terms of the transcriptome. And what we found was, so this patient is the one all the way to the right, that many genes in the type 1 interferon response were all downregulated and never responded to viral infection in this patient compared to the other patients. So basically, this patient had the most highly defective type 1 interferon pathway of all the patients, and maybe that's why what we saw happened. So the conclusions from this study were that transgenes can become a liability when things were intended for safety. So now a lot of effort went into making that virus, GMP, tox studies, and now guess what? We have to go back to the drawing board to make a better virus. Even if you inject the virus into a tumor locally, you can get it to spread to other areas because they can become viral factories. And Most importantly, we probably have to select patients very carefully as to who could have a lot of toxicity and actually who could benefit from these therapies. So I think going forward, the patients who will have the most benefit will be the ones who have a defective type 1 interferon response, and they probably shouldn't get the vector I showed you, but maybe more like a wild type of vector. And multiple types of mechanisms can be involved as opposed to just killing the cells. So future directions are what I described to you, of course, combining this with immunotherapies such as immune checkpoint inhibitors would also be warranted. I'd like to close by thanking patients and their families, which is where we start, as I described in terms of our research, all the various sources of funding that help make these things possible. And I'm, of course, presenting this work on behalf of many team members of my own group as well as collaborators. So I'll stop there and happy to take any questions.
Thank you very much. Any quick questions? And I'll ask you all to use the microphone. Anybody have any questions? Sure.
In your RNA sequencing data, were you able to sequence the virus to see if the virus had acquired any alterations in the
Some
patient?
mutations? Yeah, no, that's a good question. We were, and we were able to also isolate, I didn't show it here for interest of time, several mutant viruses, but what we found was that the viruses that had the mutations had less fitness than the original virus. So the one that persisted and did the best was the one that we had kind of started with.
Other questions?
So it looked like patient 12 had significant therapies prior. Do you think there was any evidence that that person was immunocompromised, not just in terms of interferon, but that maybe because of massive immunocompromisation that led to the outcome?
Yeah, no, that's a good question. The patient had a lot of prior therapies and actually had received radiation therapy to one of the areas about 6 months prior to the treatment, but so had all the other patients. So could this be the only factor that was different? We don't know, but given the mechanism of action, we feel it's a very likely possibility.
Other questions?
I may have missed it, but what did you find in terms of transmission? So when you treated the patient with the virus, do you think there was any risk of this virus being transmitted to others?
Yeah, very low likelihood. In all the other patients I showed you, we could not detect the virus in any fluids just a day later. Even in the last patient that I showed you where there was persistence of the virus, there was no, at least overt infection in any of the healthcare workers who came in contact with the patient and family members who came in contact with the patient. Usually we observe what are called biosafety level 2 precautions when doing this kind of work, which is the same as if a patient has HIV or hepatitis B or hepatitis C, et cetera. So nothing different compared to what's usually done in care of many patients who are in hospitals had to be done with this virus.
You mentioned that the responding patient or the acute response patient had an atypical liver cancer, that wasn't traditional liver cancer, or so what? Can you expand on that a little?
The first patient that I showed had a rare cancer called fibrolamellar hepatocellular cancer. However, I showed 3 other patients that had some level of activity, and they had the typical liver cancer. So I don't think there was anything particular about the rare one.
Patient 12 had standard hepatocellular cancer.
Actually, patient 12 had colon cancer.
Right. So
Right.
I guess that's what it was.
Right.
So do you think that had an effect on this?
Oh, could the colon cancer aspect possibly? Right. So one of the things.
Into the response because.
Right. So one of the things that a number of the groups have been working towards now is seeing, looking at TCGA data, for example, seeing what are the prevalences of defective type 1 interferon responses in different cancers. And we quite haven't arrived at an answer yet, but it seemed like in colon cancer was a bit higher. So maybe that's how we ended up with that patient in our group of 12 and where all this happened, right?
Okay, I think we're at the edge of time, so I want to thank Mitesh again. Thank you very much.